Computed axial tomography, CAT, CT scan, eye introduction. Computed axial tomography, CAT, more commonly referred to as CT scan, is a sophisticated imaging technique that utilizes X-ray technology and computer processing to create detailed cross-sectional images of the internal structures of the body. Unlike conventional X-ray imaging, which produces flat two-dimensional images, CT provides high-resolution images of slices through the body or brain, allowing a much clearer view of soft tissues, bones, blood vessels, and pathological changes. In life sciences and neuroscience, CORT is often used for detecting tumors, hemorrhages, bone fractures, and guiding biopsies. 2. Principle and mechanism. A CT scanner consists of a rotating X-ray source and a series of detectors arranged in a gantry. The patient lies on a motorized table that moves through the scanner. As the X-ray tube rotates around the patient, it emits a thin, fan-shaped beam of X-rays through the body. The detectors capture the varying amounts of X-ray energy that pass through different tissues. The key principle behind CT is that different tissues absorb X-rays to different extents depending on their density. For example, bone absorbs more X-rays than soft tissue, and soft tissue absorbs more than air. The collected data from multiple angles are processed by powerful algorithms to reconstruct cross-sectional images, tomographic slices. These slices can be compiled into a 3D representation of the scanned region. 3. Applications in biology and medicine. CT scans are extremely valuable in clinical diagnostics, particularly in neurology, oncology, and cardiology. In brain studies, CORT is commonly used in acute settings such as suspected stroke, trauma, or bleeding because it can quickly reveal hemorrhages, skull fractures, or brain swelling. It is a preferred imaging technique in emergency care due to its speed and wide availability. CT is also used in biomedical research to study animal models, internal organ structures, and bone morphology. It helps track the progression of diseases, evaluate the effects of treatments, and perform morphometric analysis. High-resolution micro-CT has been developed to study verifying structural details in small specimens, including developing embryos, tissues, and bone architecture. IV. Differences from MRI and fMRI. Unlike MRI, which relies on magnetic fields and radiofrequency signals, CT uses ionizing radiation. CT has a higher spatial resolution than MRI for detecting bony structures and calcifications but offers less contrast for soft tissues compared to MRI. It is also faster than MRI, making it suitable for imaging unstable patients or trauma victims. However, CT does not provide functional information like fMRI, as it only reflects anatomical structures and tissue density, not metabolic or blood flow changes. B. Safety and limitations. The main limitation of CT is the exposure to ionizing radiation, which can increase the risk of cancer with excessive use, especially in children and pregnant women. Contrast agents, often iodine-based, are sometimes used to enhance tissue visibility, but they may cause allergic reactions or affect kidney function in sensitive individuals. Despite these limitations, the benefits of accurate and rapid diagnosis often outweigh the risks, especially in acute clinical settings. Advances in technology, such as low-dose CT protocols, have reduced the radiation burden significantly. B. CSIR-NET Previous Year Questions 1. CSIR-NET June 2016. Which imaging technique uses X-rays and computer reconstruction to generate cross-sectional anatomical images? A. MRI. B. PET circuit CT. Answer. C. CT. Explanation. CT or CAT scan uses X-rays and computer algorithms to create detailed tomographic images. 2. CSIR-NET DEC 2020. Which of the following is most suitable for rapid imaging of brain trauma in emergency situations? A. fMRI. B. CT scan. C. PET scan. Answer. B. CT scan. Explanation. CT is preferred in emergencies due to its speed and ability to detect hemorrhage, fractures, or edema. 3. CSIR-NET June 2019. In CT scanning, contrast differences are due to a variations in magnetic field strength, the electrical activity of tissue, C. Absorption of X-rays by different tissues, the oxygenation levels in the blood. Answer. C. Absorption of X-rays by different tissues. Explanation. CT contrast arises from differential absorption of X-rays based on tissue density. Let me know if you'd like the next lecture on calcium imaging, optogenetics, EEG, or any other technique from the CSIR-NET Life Sciences syllabus. You said. Methods in field biology. ChatGPT said. Here is your CSIR-NET Life Sciences oriented high-quality theory lecture on methods in field biology, written in maximum detail, paragraph format, note tables, and including previous year CSIR-NET questions with answers and years. In field biology. Introduction. Field biology involves the direct study of organisms in their natural environment, enabling ecologists and evolutionary biologists to investigate species interactions, behavioral ecology, population dynamics, biodiversity, and ecosystem processes. Unlike laboratory-based research, field biology captures ecological realism, helping understand natural variability, adaptations, and evolutionary processes. The methodologies used in field biology are diverse and depend on the objectives of the study, the habitat, and the target species. Observational methods. A fundamental method in field biology is direct observation, which includes both continuous and time sample monitoring of behavior, movements, and interactions. These observations may be focal animal sampling, monitoring one individual, or scan sampling, scanning a group at intervals. Observational techniques are crucial in ethology, foraging studies, and territoriality assessments. The accuracy of behavioral observations often requires long-term monitoring and minimal observer interference, using blinds or hides to reduce observer effects. 3. Mark recapture techniques. Mark recapture is a widely used method to estimate population size, survival rates, and movement patterns, especially for elusive or mobile animals. Individuals are captured, marked uniquely, e.g., bands, tags, paints, and released. After a certain period, another capture is conducted, and the number of marked versus unmarked individuals is analyzed using statistical models like the Lincoln-Pearson estimator for closed populations or Jolly-Siever models for open populations. Mark recapture is extensively used in population ecology, wildlife management, and conservation biology. I. Radiotelemetry and GPS tracking. To monitor the movement, home range, and migration patterns, radiotelemetry and GPS devices are attached to animals. Radiotelemetry involves fitting animals with a transmitter that emits signals detected by a receiver. GPS collars store or transmit data on animal positions, offering high spatial resolution over large areas. These methods are crucial in spatial ecology, predator-prey dynamics, and habitat selection studies. Ethical considerations and the impact of devices on animal behavior must be carefully assessed. B. Quadrats and transects. For vegetation surveys, insect counts, and sessile organisms like coral reefs, field biologists use quadrats, fixed area sampling units, and transects lines along which sampling is conducted. These tools help assess species richness, density, abundance, and diversity indices. Random, systematic, or stratified sampling methods ensure statistical validity. In forest ecology, line intercept and point-centered quarter methods are used for tree sampling. B. Camera traps and remote sensing. Camera traps are motion-sensitive cameras deployed in natural habitats to photograph or video record animals. They are invaluable for studying nocturnal, cryptic, or rare species, allowing identification, abundance estimation, and behavioral analysis without human presence. Remote sensing techniques, using satellite imagery or drones, help map land cover, vegetation types, deforestation, and even wildlife distributions. These non-invasive methods are key in large-scale ecological monitoring and landscape ecology. 7. Environmental DNA (EDNA). A modern tool in field biology is environmental DNA analysis, where DNA fragments shed by organisms into their environment, e.g., water, soil, are collected and analyzed to detect presence or absence of species. EDNA is highly effective in studying aquatic organisms, invasive species, and biodiversity assessments in hard-to-access habitats without requiring physical capture or visual detection. 8. Bioacoustics and behavioral recordings. Bioacoustic monitoring involves recording animal vocalizations to study communication, meaning behavior, or species presence. It is commonly used in birds, bats, amphibians, and marine mammals. Software tools analyze frequency, amplitude, and temporal patterns, aiding in species identification and population density estimation. Such non-invasive methods are growing in significance for long-term monitoring in changing environments. Hicks. Ethical and statistical considerations. All field biology methods must ensure ethical treatment of animals, minimal disturbance, and appropriate permissions. Statistical rigor in sampling design, replication, and data analysis is essential for robust conclusions. Use of GIS and spatial statistics helps visualize and interpret field data effectively. CSIRNet previous year questions 1. CSIRNet June 2022. A researcher studying butterfly population size in a forest uses a mark recapture method. Which of the following assumptions must be valid for accurate estimation? Of the marked individuals die after release. B. No immigration or emigration occurs during the study. C. New individuals are marked in every recapture. D. All individuals can be caught in the first sampling answer. B. No immigration or emigration occurs during the study. Explanation. Mark recapture assumes a closed population with no changes due to migration, birth, or death. 2. CSIRNet Deck 2018. Which of the following is the best method to monitor large mammals like tigers in dense forests? A quadrat sampling. B. Pitfall trapping. C. Camera traps. D. Sweep nets. Answer. C. Camera traps. Explanation. Camera traps are ideal for monitoring large, elusive mammals in dense or inaccessible habitats. 3. CSIRNet June 2020. A scientist is using transect lines to study plant species diversity in a grassland. This method is appropriate for a. Measuring animal behavior. B. Estimating pollen viability. C. Studying vegetation distribution across gradients. D. Recording weather conditions. Answer. C. Studying vegetation distribution across gradients. Explanation. Transect sampling helps assess how species composition changes along environmental gradients. Let me know if you'd like the next lecture on statistical methods in ecology, diversity indices, habitat fragmentation studies, or any other topic from the CSIRNet Life Sciences syllabus. You said. Methods of estimating population density of animals. ChatGPT said. Here is your CSIRNet Life Sciences oriented detailed theory lecture on methods of estimating population density of animals with no tables, written in high quality paragraph form, and including CSIRNet previous year questions with answers in years. Methods of estimating population density of animals by introduction. Population density refers to the number of individuals of a species per unit area or volume and is a key parameter in population ecology. Estimating the density of animal populations is crucial for understanding population dynamics, species interactions, resource use, conservation status, and ecological modeling. The methods used vary depending on the mobility, visibility, habitat, and size of the organisms. Broadly, estimation techniques include direct counts, indirect indices, capture mark recapture, and distance sampling, each with specific assumptions and limitations. 2. Direct count methods. Direct count or total enumeration is the most straightforward method and involves physically counting all individuals within a defined area. This method works best in small populations, open habitats, or for sessile or slow-moving organisms such as mollusks, amphibians during breeding congregations, or colony nesting birds. However, it becomes impractical or inaccurate for large, cryptic, or mobile populations. Aerial surveys, boat-based counts, or ground transects are used to directly observe and count large animals like elephants or whales. 3. Sampling and extrapolation. When total counts are not feasible, plot-based sampling, e.g., quadrats or transects, is used. A number of small, representative areas are sampled, and the counts are extrapolated to estimate the population density in the entire study area. This technique assumes that the sample plots are representative of the habitat as a whole. For mobile animals, line transect sampling is widely used, wherein observers record animals encountered along defined lines and use statistical models to estimate density. IV. Mark recapture method. For mobile and elusive animals, the capture mark recapture (CMR) technique is the most commonly used method to estimate density. It involves capturing individuals, marking them with tags, bands, or paints, and releasing them back into the population. After a specific period, a second capture is made, and the ratio of marked to unmarked individuals is used to estimate population size using models like the Lincoln-Pearson estimator for closed populations or Jolly-Sieber models for open populations. This method requires that marks are not lost, that animals are not behaviorally affected by capture, and that the population remains closed during the study. B. Indirect methods and indices. When animals are difficult to detect directly, indirect signs such as fecal droppings, tracks, nests, burrows, or calls can be used as indices of density. These index-based methods assume a correlation between the index and actual density, which must be calibrated through field validation. Such methods are widely used in mammalogy, e.g., tiger pug marks, ornithology, bird call counts, and entomology, trap counts. B. Distance sampling and detection probability. Distance sampling techniques, such as line transect and point transect surveys, involve recording the distance of each detected individual from the observer. These distances are used to model the detection function, which accounts for the fact that animals farther from the observer are less likely to be seen. This approach helps estimate true density while correcting for detectability. This method is especially valuable in bird and mammal surveys in forests and grasslands. 7. Camera trap-based capture recapture. For cryptic and nocturnal species, camera trap-based mark recapture methods are used. Animals are identified by natural markings, like stripes or spots, or radio collars. This method has revolutionized density estimation in big cats, bears, and ungulates, particularly in dense habitats where direct observation is limited. Statistical models such as spatially explicit capture recapture (SECR) provide more accurate density estimates by incorporating spatial information from detections. 8. Aerial and acoustic surveys. Aerial surveys using drones, planes, or helicopters are used to count large-bodied animals in open landscapes, such as antelopes, seals, or whales. Acoustic surveys, using call playback or sound recording equipment, are applied in birds, frogs, cetaceans, and bats. These methods rely on known call rates or signal detection probabilities for density estimation. Hicks. Environmental DNA (EDNA). EDNA sampling involves collecting water or soil samples and analyzing them for DNA fragments shed by animals. While currently more suited for presence-absence data, developments are underway to quantify population densities using EDNA concentration calibrated with experimental data. It offers a non-invasive way to monitor aquatic and rare species. XCSIRNet previous year questions 1. CSIRNet Deck 2018. Which of the following methods is best suited for estimating population density of elusive large carnivores like tigers in a forest? A sweep netting. B. Camera trap-based capture recapture C. Quadrats. D. Pitfall traps. Answer. B. Camera trap-based capture recapture. Explanation. Camera traps allow for non-invasive individual identification and recapture analysis. 2. CSIRNet June 2017. In a capture mark recapture study, which of the following assumptions is critical for accurate population density estimation? All individuals can be caught only once. B. Marked individuals mix randomly with the population. C. Population increases during the study. D. Animals forget being captured. Answer. B. Marked individuals mix randomly with the population. Explanation. This ensures equal catchability and unbiased estimates. 3. CSIRNet Deck 2020 line transect sampling is used to estimate allele frequencies. B. Estimate species richness. C. Estimate animal density while correcting for detectability. D. Measure plant growth. Answer. C. Estimate animal density while correcting for detectability. Explanation. Line transects record detections and distances to estimate true density. Let me know if you'd like the next lecture on population growth models, life tables, species interactions, or any other topic from CSIRNet Life Sciences syllabus. You said. A plant's ChatGPT said. Here is your CSIRNet Life Sciences oriented theory lecture on methods of estimating population density of plants, written in detailed paragraph form, with no tables, and including previous year CSIRNet questions with answers in here. This lecture is structured to serve as high-quality core material for your exam. Methods of estimating population density of plants. Introduction. Estimating population density in plant ecology is a foundational method used to understand species distribution, community structure, competition, productivity, and the impact of environmental variables. Unlike animals, plants are generally immobile and easier to count, but their density estimation must consider size variation, clonality, growth habit, and habitat heterogeneity. The most widely used methods include quadrats, transects, plotless sampling techniques, and remote sensing approaches, each selected based on the ecological context and plant characteristics. 2. Quadrat sampling. The quadrat method is the most common approach to estimate plant population density. A quadrat is a square or rectangular frame of known size that is placed randomly or systematically across the study site. All individual plants within the quadrat are counted, and the average count per unit area is used to estimate density per square meter or hectare. This method is best suited for herbaceous plants, grasses, and ground cover species. Quadrat size must be appropriate for the plant type, smaller quadrats for smaller species and larger ones for shrubs and saplings. In random sampling, quadrats are placed randomly to avoid bias. In systematic sampling, quadrats are placed at regular intervals, which is efficient but maintenance patchy distributions. Stratified sampling is used in heterogeneous habitats where each stratum is sampled proportionally. 3. Line and belt transects. Line transects involve stretching a line like a measuring tape or string across a habitat and recording plant individuals that touch or intersect the line. This is particularly useful in habitats with linear gradients such as along slopes, wetland edges, or coastal dunes. Belt transects are broader and involve sampling a strip of known width along the line. All individuals within the belt are counted. Methods of estimating population density of plants. Introduction. Estimating population density in plant ecology is a foundational method used to understand species distribution, community structure, competition, productivity, and the impact of environmental variables. Unlike animals, plants are generally immobile and easier to count, but their density estimation must consider size variation, clonality, growth habit, and habitat heterogeneity. The most widely used methods include quadrats, transects, plotless sampling techniques, and remote sensing approaches, each selected based on the ecological context and plant characteristics. 2. Quadrat sampling. The quadrat method is the most common approach to estimate plant population density. A quadrat is a square or rectangular frame of known size that is placed randomly or systematically across the study site. All individual plants within the quadrat are counted, and the average count per unit area is used to estimate density per square meter or hectare. This method is best suited for herbaceous plants, grasses, and ground cover species. Quadrat size must be appropriate for the plant type, smaller quadrats for smaller species and larger ones for shrubs and saplings. In random sampling, quadrats are placed randomly to avoid bias. In systematic sampling, quadrats are placed at regular intervals, which is efficient but maintenance patchy distributions. Stratified sampling is used in heterogeneous habitats where each stratum is sampled proportionally. 3. Line and belt transects. Line transects involve stretching a line like a measuring tape or string across a habitat and recording plant individuals that touch or intersect the line. This is particularly useful in habitats with linear gradients such as along slopes, wetland edges, or coastal dunes. Belt transects are broader and involve sampling a strip of known width along the line. All individuals within the belt are counted, providing both density and spatial distribution information. Belt transects are ideal for estimating density and species richness in forest understory, successional plots, and edge habitats. IV. Point-centered quarter method (PCQM). This plotless sampling method is used for estimating density of woody plants like trees and shrubs in forests. At each sampling methods of estimating population density of plants. Introduction. Estimating population density in plant ecology is a foundational method used to understand species distribution, community structure, competition, productivity, and the impact of environmental variables. Unlike animals, plants are generally immobile and easier to count, but their density estimation must consider size variation, clonality, growth habit, and habitat heterogeneity. The most widely used methods include quadrats, transects, plotless sampling techniques, and remote sensing approaches, each selected based on the ecological context and plant characteristics. 2. Quadrat sampling. The quadrat method is the most common approach to estimate plant population density. A quadrat is a square or rectangular frame of known size that is placed randomly or systematically across the study site. All individual plants within the quadrat are counted, and the average count per unit area is used to estimate density per square meter or hectare. This method is best suited for herbaceous plants, grasses, and ground cover species. Quadrat size must be appropriate for the plant type, smaller quadrats for smaller species and larger ones for shrubs and saplings. In random sampling, quadrats are placed randomly to avoid bias. In systematic sampling, quadrats are placed at regular intervals, which is efficient but maintenance patchy distributions. Stratified sampling is used in heterogeneous habitats where each stratum is sampled proportionally. 3. Line and belt transects. Line transects involve stretching a line like a measuring tape or string across a habitat and recording plant individuals that touch or intersect the line. This is particularly useful in habitats with linear gradients such as along slopes, wetland edges, or coastal dunes. Belt transects are broader and involve sampling a strip of known width along the line. All individuals within the belt are counted, providing both density and spatial distribution information. Belt transects are ideal for estimating density and species richness in forest understory, successional plots, and edge habitats. IV. Point-centered quarter method (PCQM). This plotless sampling method is used for estimating density of woody plants like trees and shrubs in forests. At each sampling point, the area is divided into 4 quadrants, and the nearest individual plant in each quadrant is measured for distance. These distances are used to estimate average spacing and, consequently, plant density using mathematical models. PCQM is highly effective in dense forests, tropical woodlands, and areas where establishing large plots is impractical. However, it assumes random distribution, and deviations from this assumption can introduce bias. V. Remote sensing and GIS-based estimation. With the advent of remote sensing, satellite imagery and drone-based aerial photography are used to estimate plant population density over large areas, particularly for agricultural crops, forests, and invasive species. Vegetation indices such as NDVI, normalized difference vegetation index, correlate with plant biomass and coverage and can serve as proxies for density. Ground truthing is necessary to calibrate remote data with actual field measurements. B. Seedling counts and germination plots. For annuals and ephemeral plants, especially in desert or disturbed habitats, density estimation is done by marking plots during germination and counting seedlings. This provides a more accurate picture of reproductive output and regeneration. In long-term ecological studies, permanent plots are maintained to monitor changes in plant density over time. 7. Challenges and considerations. Estimating plant density involves challenges such as overlapping individuals, clonal growth, and cryptic species. For clonal species like grasses, density estimation must distinguish between remettes, physically separate units, and genets, genetic individuals, as counting remettes may overestimate density. Similarly, seasonal variation, observer bias, and sampling error must be minimized through replication and standardized protocols. 8. CSIR-NET previous year questions 1. CSIR-NET June 2015. Which of the following methods is most suitable for estimating the density of herbaceous plant species in a grassland? A point count method. B. Quadrat sampling. C. Capture mark recapture. D. Radiotelemetry. Answer. B. Quadrat sampling. Explanation. Quadrats are widely used for density estimation in herbaceous communities like grasslands. 2. CSIR-NET Deck 2019. In the point-centered quarter method, what is primarily measured at each sampling point? A number of flowers. B biomass of the plant. C distance to nearest plant in each quadrant. D height of tallest plant. Answer. C distance to nearest plant in each quadrant. Explanation. PCQM estimates density by averaging distances to nearest individuals in 4 quadrants. 3. CSIR net June 2017. Which one of the following methods is best suited for estimating density of canopy trees in a dense tropical forest without laying plots? A strict transect. B quadrat C line transect. D point centered quarter method. Answer. D point centered quarter method. Explanation. PCQM is plotless and efficient for estimating tree density in dense vegetation. Let me know if you'd like the next lecture on diversity estimation methods, species area relationships, or any other part of the CSIR net ecology syllabus. You said, ranging patterns through direct observations. ChatGPT said. Here is your CSIR net life sciences oriented detailed theory lecture on ranging patterns through direct observations, written in high quality paragraph form with no tables and including CSIR net previous year questions with answers in here. This is part of your field biology method series. Ranging patterns through direct observations. Introduction. Ranging patterns refer to the spatial movement behavior of animals within their home ranges or territories over time. Understanding these patterns is fundamental to animal ecology and behavior, as they reveal information about resource use, social structure, territoriality, habitat preferences, and energetics. Direct observation is one of the oldest and most informative methods for studying ranging patterns in the wild. It involves visually tracking animals and recording their locations, movements, and behaviors without the use of automated devices. 2. Principles of ranging and home range. A home range is the area in which an individual or group regularly moves to find food, mates, and shelter. It is not defended, unlike a territory, which is aggressively protected from others. Ranging behavior includes the daily or seasonal movements within these areas. It is influenced by several ecological factors such as resource distribution, population density, predation risk, reproductive cycles, and group size. 3. Direct observation techniques. In direct observation, trained observers follow focal animals or groups, either on foot or using vehicles, and record their positions at regular intervals. This is often done using compasses, maps, GPS devices, or triangulation methods to plot the animal's location. The observer keeps a safe distance to avoid influencing the animal's behavior while maintaining a visual contact to accurately record its movements. Animals are followed over extended periods, and their location data is used to map their paths, estimate range size, and understand movement patterns such as daily paths, resting spots, and core areas of activity. IV. Focal and scan sampling in ranging studies and direct observations. 2 sampling strategies are typically employed. Focal animal sampling involves continuous observation of one individual or group for a fixed period. This method is excellent for detailed behavioral and spatial data, including time spent in specific areas, types of activities, and interaction with landscape features. Scan sampling involves recording the location and behavior of all visible individuals at fixed time intervals. Though less detailed, this method allows for broader coverage of movement within groups. Mapping and data analysis. The recorded positions are plotted on maps to visualize the ranging area, which can be further analyzed using methods like minimum convex polygon (MCP) encloses all recorded points with the smallest possible convex polygon. It gives a rough estimate of home range size but can be sensitive to outliers. Kernel density estimation (KDE), a more refined method that calculates the intensity of use across space and identifies core areas where animals spend most of their time and peripheral zones. Path analysis tracks are analyzed to assess distance traveled, speed, turning angles, and resting periods. B. Advantages and limitations. Direct observation provides rich, behaviorally contextualized data and allows researchers to observe real-time decisions, social interactions, and habitat use. However, it is labor-intensive, time-consuming, and often limited to diurnal and habituated species in open or semi-open habitats. It is less effective for nocturnal, cryptic, or arboreal animals in dense forests, and less special methods like night vision or canopy access are used. 7. Ethical and practical considerations. Long-term direct observation demands minimal interference, non-invasive techniques, and observer habituation to avoid altering animal behavior. In some cases, radio coloring, color bands, or individual markings may be used to reliably identify individuals during tracking. 8. Integration with other methods. While direct observation alone can offer deep insight, it is often combined with GPS telemetry, camera traps, or drones for more comprehensive spatial and temporal coverage. This multi-method approach strengthens the accuracy and ecological interpretation of ranging behavior. Hicks. CSIR net previous year questions 1. CSIR net deck 2016. A researcher wants to study the home range and core area of a primate group using direct observation. Which of the following techniques is most appropriate to estimate core areas used more intensively? A minimum convex polygon. B kernel density estimation. C point centered quarter method. D line transects. Answer. B kernel density estimation. Explanation. KDE provides a probabilistic estimate of space use, highlighting core areas. 2. CSIR net June 2018. Which method is most suited for assessing ranging behavior of diurnal, terrestrial mammals in open habitats? A scan sampling combined with GPS mapping. B pitfall traps. C aerial surveys. D capture mark recapture. Answer. A scan sampling combined with GPS mapping. Explanation. This provides systematic spatial data for visible mammals during the day. 3. CSIR net deck 2021. Which of the following statements is true about ranging behavior in animals? A home range is always smaller than the territory. B territory is never defended. C all ranging is random and unpredictable. D core areas are used more intensively within the home range. Answer. D core areas are used more intensively within the home range. Explanation. Core areas represent frequent use zones, often including feeding or nesting sites. Let me know if you want the next lecture on radio telemetry, home range estimation models, or habitat preference studies. You said. Indirect ChatGPT said. Here is your CSIR net life sciences oriented detailed theory lecture on ranging patterns through indirect methods, written in a high quality, paragraph style format with no tables, and including CSIR net previous year questions with answers in here. This is a continuation of the topic on methods in field biology. Ranging patterns through indirect methods. Introduction. Indirect methods for studying ranging patterns are essential tools in field biology, especially when direct observation of animals is not feasible. These methods rely on signs, remains, or recordings left by animals to infer their movements, home ranges, and habitat use. They are particularly valuable for studying nocturnal, cryptic, solitary, or dangerous species that are difficult to follow visually. Indirect methods often complement direct observation, providing a broader and sometimes longer-term view of spatial behavior. 2. Types of indirect methods. 1. Animal signs and traces. Tracking animals through feces, scats, footprints, scratches, nests, burrows, feeding remains, and urine marks is one of the most traditional indirect methods. Repeated presence of these signs in specific areas indicates frequent usage and helps map ranging boundaries and movement corridors. For example, large carnivores often use scent marking or scrape marks to delineate territories, and these signs can be used to estimate home range size and overlap with other individuals. 2. Camera traps. Camera traps are motion or heat-triggered cameras placed in the field that automatically photograph or record animals when they pass by. When multiple cameras are strategically placed, they can provide information on movement patterns, site fidelity, and temporal activity patterns. By analyzing the frequency of appearances and locations of sightings, ecologists can infer ranging patterns over time. These devices are particularly useful in dense forests, mountainous terrains, and for studying rare or nocturnal species. 3. Radio telemetry and GPS collars as indirect support. Though not purely indirect, these methods use automated location data collected through radio signals or GPS satellites. When animals cannot be visually followed, the transmitted coordinates can be used to map their range, daily paths, and preferred zones. In such cases, the movement is not seen directly by the observer but is inferred from remote data, hence considered an indirect observation tool in many ecological studies. 4. Nest or den monitoring. For species that regularly return to nests, burrows, or roosting sites, monitoring these locations can yield insights into their ranging behavior. The frequency and timing of returns can indicate central place foraging, while changes in nest sites may reflect seasonal or resource-based movement patterns. 5. DNA and hair traps. Non-invasive sampling using feces, urine, or hair collected from the field can be used for genetic identification of individuals. When repeated samples from the same individual are found in different locations, researchers can infer movement ranges and spatial overlap. These methods are especially useful in large, elusive mammals such as tigers, wolves, and bears. 3. Data interpretation and mapping. All indirect data are georeferenced and plotted using GIS, geographic information systems, or spatial software to produce maps of movement paths, range size, and site preference. Analytical tools such as minimum convex polygon (MCP) and kernel density estimation (KDD), though more commonly used with direct tracking data, can also be applied to presence records from indirect methods. Temporal data timestamps from camera traps, for example, further enhance analysis by showing activity rhythms. IV. Advantages and limitations. Indirect methods are less invasive, suitable for long-term monitoring, and can cover large areas with minimal manpower. They are essential for studying endangered or sensitive species where disturbance must be minimized. However, they often suffer from lack of behavioral context, observer interpretation bias, and difficulty in individual identification unless combined with genetic tagging or photo recognition. These methods also depend heavily on correct identification of signs, which requires training and expertise. V. Integration with other methods. Indirect methods are most powerful when integrated with direct observations, ecological modeling, and remote sensing. For instance, camera trap data can be cross-verified with DNA analysis for individual ID, or movement inferred from signs can be modeled in relation to landscape features using GIS. B. CSIRNet previous year questions 1. CSIRNet Deck 2017. Which of the following is an indirect method for studying ranging behavior of elusive mammals? A direct observation from blinds. B. Radio collar tracking. C. Analyzing footprints and feces. D. Focal animal sampling. Answer. C. Analyzing footprints and feces. Explanation. Tracking signs like scats and prints is an indirect way to estimate movement and presence. 2. CSIRNet June 2021. Camera traps are particularly useful in studying which of the following: A. Animal coloration in the wild. B. Photosynthetic efficiency. C. Ranging patterns and activity schedules of nocturnal animals. D. Feeding rates in insects. Answer. C. Ranging patterns and activity schedules of nocturnal animals. Explanation. Camera traps provide timestamp data on movements without direct observation. 3. CSIRNet Deck 2016. Which method would be least invasive for estimating the home range of a threatened tiger population? A. Tagging with GPS collars. B. Aerial tracking. C. Hair and fecal DNA sampling. D. Immobilizing and attaching radio transmitters. Answer. C. Hair and fecal DNA sampling. Explanation. Non-invasive genetic sampling avoids handling animals and gives spatial data. Let me know if you want the next lecture on radio telemetry, home range estimation models, or any other field ecology or behavioral technique for CSIRNet. You said, and remote ChatGPT said. Here is your CSIRNet life sciences oriented high quality theory lecture on ranging patterns through remote methods, with no tables, written in detailed paragraph style, and including previous year CSIRNet questions with answers in here. This lecture is part of the series under methods in field biology. Ranging patterns through remote methods. Introduction. Remote methods for studying ranging patterns refer to automated, technologically mediated techniques that track animal movements from a distance, often without requiring continuous human presence. These methods are particularly valuable in challenging terrains, dense forests, or for nocturnal, highly mobile, or elusive species. They allow researchers to gather long-term, high-resolution spatial data, sometimes at scales unachievable through direct or indirect observation. Remote sensing has revolutionized wildlife ecology by facilitating precise tracking, real-time data acquisition, and integration with ecological models. 2. Radio telemetry. One of the earliest remote methods used in animal tracking, radio telemetry, involves attaching a small radio transmitter to an animal via collars, harnesses, or implants. Each transmitter emits a unique radio frequency that can be picked up by a handheld or automated receiver. Researchers use directional antennas to triangulate the position of the animal by moving through the landscape and determining the direction and strength of the signal. This method provides moderately precise location data and is suitable for short to medium range tracking in both terrestrial and avian species. 3. GPS tracking systems. More advanced than radio telemetry, GPS collars or tags use satellite-based global positioning systems to record animal locations at fixed time intervals. The data can either be stored onboard for later retrieval or transmitted remotely via GSM, mobile network, or satellite uplinks, e.g., Argos system. GPS tracking offers high spatial and temporal resolution, allowing the study of movement paths, home ranges, migration routes, and habitat preferences over time. It is widely used in large mammals, sea turtles, birds, and even flying insects. IV. Satellite remote sensing. In some cases, the animals themselves are not tagged, but instead habitat changes and movement corridors are inferred using satellite imagery and remote sensing tools. For example, changes in vegetation, water availability, or fire events may influence animal ranging patterns. Integrating animal tracking data with habitat maps from satellites like Landsat, MODIS, or Sentinel enables ecologists to understand how animals respond to landscape level changes. The acoustic telemetry. Particularly useful in aquatic environments, acoustic telemetry involves the attachment of underwater transmitters that emit sound pulses. These signals are detected by hydrophone receivers placed at strategic locations, enabling tracking of fish or marine mammals. The system provides movement information within the range of hydrophones and is effective in oceans, rivers, or lakes where GPS cannot be used. B. Drone-based tracking. Unmanned aerial vehicles, UAVs, or drones, are increasingly used in wildlife studies to visually track animals from the sky. They can be fitted with thermal imaging, GPS, or camera systems, allowing detection of animals from above without disturbing them. Drones are especially useful for large-scale surveys, nest mapping, or tracking herd movements across open landscapes. 7. Advantages of remote methods. Remote tracking methods provide objective, quantitative, and continuous data on animal movement. They reduce observer bias, can be applied in inaccessible habitats, and allow multi-individual monitoring over large geographic areas. The timestamp data also allows researchers to analyze daily activity rhythms, seasonal movements, responses to disturbances, and territorial behavior. 8. Challenges and limitations. Despite their power, remote methods come with certain limitations. High costs, equipment failures, data gaps, battery limitations, and ethical concerns related to animal tagging must be considered. In some habitats, such as dense forests or deep oceans, signal transmission may be unreliable. Proper data analysis requires advanced software, GIS expertise, and ecological modeling skills. X. Integration with other methods. The best results are often achieved by combining remote methods with direct observation, habitat assessments, or genetic tools. For example, GPS data can be combined with satellite vegetation maps to infer resource selection, or movement data can be analyzed alongside behavioral observations to interpret ecological strategies. XCSIRNet previous year questions 1. CSIRNet Deck 2019. Which of the following methods would best provide high resolution, timestamp data on animal movement across large spatial scales? A direct focal sampling. B. Radio telemetry. C. GPS collars with satellite uplink. D. Hair trap analysis. Answer. C. GPS collars with satellite uplink. Explanation. GPS collars allow precise, continuous tracking and remote data access. 2. CSIRNet June 2022. Which technique is most suitable for tracking the underwater movement of marine animals? A. GPS telemetry. B. Aerial drone. C. Acoustic telemetry. D. Central analysis. Answer. C. Acoustic telemetry. Explanation. GPS is ineffective underwater. Acoustic tags work well with hydrophones. 3. CSIRNet Deck 2018. In remote tracking of terrestrial mammals, which factor is least likely to interfere with GPS signal transmission? A dense forest canopy. B. Burrowing behavior. C. Open grasslands. D. Deep caves. Answer. C. Open grasslands. Explanation. Open areas allow uninterrupted satellite signal reception. Let me know if you want the next lecture on home range estimation models, habitat preference quantification, or mark recapture methods for CSIRNet. You said. Sampling methods in the study of behavior. ChatGPT said. Here is your CSIRNet Life Sciences oriented high-quality lecture on sampling methods in the study of behavior, written in detailed paragraph-style format with no tables and including CSIRNet previous year questions with answers in year mentioned. Sampling methods in the study of behavior. Introduction. Behavioral studies require precise, consistent, and systematic sampling of observed actions or responses in animals or humans. Sampling methods determine how, when, and what behaviors are recorded during observation. Ranging patterns through remote methods. Introduction. Remote methods for studying ranging patterns refer to automated, technologically mediated techniques that track animal movements from a distance, often without requiring continuous human presence. These methods are particularly valuable in challenging terrains, dense forests, or for nocturnal, highly mobile, or elusive species. They allow researchers to gather long-term, high-resolution spatial data, sometimes at scales unachievable through direct or indirect observation. Remote sensing has revolutionized wildlife ecology by facilitating precise tracking, real-time data acquisition, and integration with ecological models. 2. Radiotelemetry. One of the earliest remote methods used in animal tracking, radiotelemetry involves attaching a small radio transmitter to an animal via collars, harnesses, or implants. Each transmitter emits a unique radio frequency that can be picked up by a handheld or automated receiver. Researchers use directional antennas to triangulate the position of the animal by moving through the landscape and determining the direction and strength of the signal. This method provides moderately precise location data and is suitable for short to medium range tracking in both terrestrial and avian species. 3. GPS tracking systems. More advanced than radiotelemetry, GPS collars or tags use satellite-based global positioning systems to record animal locations at fixed time intervals. The data can either be stored on board for later retrieval or transmitted remotely via GSM, mobile network, or satellite uplinks, e.g., Argos system. GPS tracking offers high spatial and temporal resolution, allowing the study of movement paths, home ranges, migration routes, and habitat preferences over time. It is widely used in large mammals, sea turtles, birds, and even flying insects. IV. Satellite remote sensing. In some cases, the animals themselves are not tagged, but instead habitat changes and movement corridors are inferred using satellite imagery and remote sensing tools. For example, changes in vegetation, water availability, or fire events may influence animal ranging patterns. Integrating animal tracking data with habitat maps from satellites like Landsat, MODIS, or Sentinel enables ecologists to understand how animals respond to landscape-level changes. Via acoustic telemetry. Particularly useful in aquatic environments, acoustic telemetry involves the attachment of underwater transmitters that emit sound pulses. These signals are detected by hydrophone receivers placed at strategic locations, enabling tracking of fish or marine mammals. The system provides movement information within the range of hydrophones and is effective in oceans, rivers, or lakes where GPS cannot be used. B. Drone-based tracking. Unmanned aerial vehicles, UAVs, or drones, are increasingly used in wildlife studies to visually track animals from the sky. They can be fitted with thermal imaging, GPS, or camera systems, allowing detection of animals from above without disturbing them. Drones are especially useful for large-scale surveys, nest mapping, or tracking herd movements across open landscapes. 7. Advantages of remote methods. Remote tracking methods provide objective, quantitative, and continuous data on animal movement. They reduce observer bias, can be applied in inaccessible habitats, and allow multi-individual monitoring over large geographic areas. The timestamp data also allows researchers to analyze daily activity rhythms, seasonal movements, responses to disturbances, and territorial behavior. 8. Challenges and limitations. Despite their power, remote methods come with certain limitations. High costs, equipment failures, data gaps, battery limitations, and ethical concerns related to animal tagging must be considered. In some habitats, such as dense forests or deep oceans, signal transmission may be unreliable. Proper data analysis requires advanced software, GIS expertise, and ecological modeling skills. X. Integration with other methods. The best results are often achieved by combining remote methods with direct observation, habitat assessments, or genetic tools. For example, GPS data can be combined with satellite vegetation maps to infer resource selection, or movement data can be analyzed alongside behavioral observations to interpret ecological strategies. XCSIRNet previous year questions 1. CSIRNet Deck 2019. Which of the following methods would best provide high resolution, timestamp data on animal movement across large spatial scales? A direct focal sampling. B. Radiotelemetry. C. GPS collars with satellite uplink. D. Hair trap analysis. Answer. C. GPS collars with satellite uplink. Explanation. GPS collars allow precise, continuous tracking and remote data access. 2. CSIRNet June 2022. Which technique is most suitable for tracking the underwater movement of marine animals? A. GPS telemetry. B. Aerial drone. C. Acoustic telemetry. D. Central analysis. Answer. C. Acoustic telemetry. Explanation. GPS is ineffective underwater. Acoustic tags work well with hydrophones. 3. CSIRNet Deck 2018. In remote tracking of terrestrial mammals, which factor is least likely to interfere with GPS signal transmission? A dense forest canopy. B. Burrowing behavior. C. Open grasslands. D. Deep caves. Answer. C. Open grasslands. Explanation. Open areas allow uninterrupted satellite signal reception. Let me know if you want the next lecture on home range estimation models, habitat preference quantification, or mark recapture methods for CSIRNet. You said. Sampling methods in the study of behavior. ChatGPT said. Here is your CSIRNet Life Sciences oriented high-quality lecture on sampling methods in the study of behavior, written in detailed paragraph-style format with no tables and including CSIRNet previous year questions with answers in year mentioned. Sampling methods in the study of behavior. Introduction. Behavioral studies require precise, consistent, and systematic sampling of observed actions or responses in animals or humans. Sampling methods determine how, when, and what behaviors are recorded during observation. These techniques are foundational to ethology, behavioral ecology, and neuroethology, where reliable data collection is critical for understanding behavioral patterns, social dynamics, meaning systems, communication, learning, and cognition. The choice of sampling method depends on the research question, species studied, type of behavior, time constraints, and environmental conditions. 2. Major types of sampling methods. 1. Ad libitum sampling. In this method, the observer records any behavior that seems interesting or relative whenever it occurs. It is unsystematic and subjective but useful during preliminary observations to identify key behaviors or to create ethograms. However, it is prone to observer bias, and conspicuous behaviors are more likely to be recorded than subtle or rare ones. Ad libitum sampling is usually not suitable for quantitative analyses due to its lack of structure. 2. Focal animal sampling. Focal sampling involves observing one individual, the focal subject, intensively for a specified period and recording all its behaviors. This method is particularly useful when detailed data on individual activity budgets, sequence of behaviors, or individual differences are required. It allows for quantitative analysis and is ideal when marked or easily identifiable individuals are available. The observer needs to maintain continuous visual contact to ensure no behavior is missed. 3. Scan sampling. In scan sampling, the observer scans a group of animals at regular intervals, e.g., every 30 seconds or 5 minutes, and records the behavior each animal is exhibiting at that instant. This method is efficient for group-level behavior analysis, such as herd grazing, social spacing, or activity patterns. However, it may miss short duration or rare behaviors due to its interval-based nature. Scan sampling provides data on activity budgets and time allocation across group members. 4. Behavior sampling. Behavior sampling involves recording every occurrence of a specific behavior, e.g., aggression, grooming, copulation across the entire group, regardless of which individual performs it. This method is ideal for rare, critical, or socially important behaviors that occur infrequently but have high biological significance. For example, in studies of dominance, mating behavior, or conflict resolution, behavior sampling helps gather sufficient instances for analysis. 5. Continuous recording. This is the most detailed sampling method where all behaviors are recorded as they occur, with exact time of initiation and termination. It provides data on the sequence, frequency, and duration of each behavior. Though data-rich and highly accurate, it is labor-intensive and typically limited to short observation sessions or used in conjunction with focal animal sampling. 3. Choosing the right sampling method. The selection of an appropriate sampling method should align with the objective of the study. For instance, to compare dominance interactions across individuals, behavior sampling of aggressive acts is preferred. To study an animal's daily activity budget, scan sampling or focal animal sampling works well. When testing hypotheses about behavioral sequences or decision-making, continuous recording provides the necessary detail. IV. Considerations and challenges. Observer presence, observer bias, environmental visibility, and species-specific behavioral rates can all influence data quality. Use of video recordings, blind observations, and behavioral coding software like Boris, Ethovision, or Observer XT can minimize bias and enhance accuracy. Sampling methods must also account for temporal variation, day versus night behavior, habitat complexity, and inter-observer reliability. BCSIR Net Previous Year Questions 1. CSIR Net June 2016. A researcher studying the grooming behavior of langurs follows one individual at a time for 10 minutes and records all its behaviors. What sampling method is being used? Ad libitum sampling. B scan sampling, C focal animal sampling, D behavior sampling. Answer: C focal animal sampling. Explanation: the observer follows a single individual and records all behaviors, which is focal animal sampling. 2. CSIR Net Deck 2019. Which of the following sampling methods is most appropriate to quantify the frequency of a rare aggressive behavior in a troop of monkeys? A scan sampling. B ad libitum sampling, C focal animal sampling, D behavior sampling. Answer: D behavior sampling. Explanation: behavior sampling is best for capturing all instances of rare socially significant actions like aggression. 3. CSIR Net June 2021. In a study of feeding behavioral herds, an observer records what each individual is doing at fixed intervals of 5 minutes. This method is known as a focal animal sampling. B behavior sampling, C continuous recording, D scan sampling. Answer: D scan sampling. Explanation: at fixed intervals, the behavior of each group member is recorded, indicating scan sampling. Let me know if you'd like the next lecture on experimental designs in behavioral studies, measuring behavioral frequencies, or coding behavior for statistical analysis in CSIR Net Life Sciences. You said. Habitat characterization. ChatGPT said. Here is your CSIR Net Life Sciences oriented, high-quality theory lecture on habitat characterization, written in detailed paragraph style with no tables, and including CSIR Net previous year questions and answers with year mentioned. This lecture is part of the series under methods in field biology. Habitat characterization. Introduction. Habitat characterization is the process of systematically describing the physical, biological, and chemical components of a habitat where an organism or population resides. It provides essential context for understanding species distributions, population dynamics, community structure, and ecosystem functioning. Characterizing habitats is a critical first step in ecological field studies, biodiversity assessments, habitat restoration, and conservation planning. It enables researchers to quantify environmental heterogeneity and relate species presence or behavior to specific habitat features. 2. Components of habitat characterization. Habitat characterization involves identifying and measuring a range of abiotic and biotic variables that define the structure and quality of a habitat. These typically include topographic features, elevation, slope, aspect, and landscape position are recorded, often using GPS and altimeters. These factors influence microclimate and water drainage patterns. Climatic parameters, temperature, humidity, rainfall, light intensity, and wind speed are key variables monitored using meteorological equipment or data loggers. These parameters help assess the suitability of habitat conditions for different species. Soil properties, soil texture, pH, organic matter content, moisture, and nutrient availability are determined through field sampling and laboratory analysis. Soil characteristics directly affect plant communities and indirectly influence animal distribution. Vegetation structure. This includes measurements of canopy cover, vegetation density, basal area, understory composition, and plant species diversity. Quadrats and transects are commonly used for this purpose. Vegetation forms structural basis for terrestrial habitats and provides food, shelter, and breeding sites for animals. Water resources, presence, quality, and seasonal availability of water bodies, streams, ponds, wetlands are important for aquatic and semi-aquatic species. Parameters such as pH, dissolved oxygen, turbidity, and conductivity are recorded. Microhabitat features. These include elements like fallen logs, leaf litter depth, rock crevices, or burrows, which provide critical microclimates or nesting sites for many organisms, especially amphibians, reptiles, and invertebrates. Anthropogenic factors. Human disturbances such as habitat fragmentation, pollution, grazing, logging, and proximity to roads or settlements are documented to evaluate habitat degradation or modification. 3. Methods used in habitat characterization. Habitat characterization is typically conducted through field sampling, GIS and remote sensing, and environmental monitoring tools. Field sampling techniques, quadrats, line intercepts, belt transects, and point intercept methods are used to estimate vegetation composition and structure. Soil and water samples are collected for laboratory analysis. Remote sensing and GIS, satellite imagery and aerial photographs are used to map large-scale habitat features like forest cover, fragmentation, land use patterns, and vegetation indices like NDVI. These are integrated into geographic information systems, GIS, to create habitat maps and spatial models. Instrumental monitoring. Devices like soil moisture sensors, temperature loggers, and light meters help monitor environmental conditions over time. Automatic weather stations provide continuous climatic data. B. Applications of habitat characterization. Habitat characterization is crucial in ecological niche modeling, defining the environmental envelope where a species can survive. Habitat suitability index, cheat models, used in wildlife management to evaluate quality and availability of critical habitats. Conservation planning, prioritizing areas for protection or restoration based on habitat quality and threats. Species distribution models, SDMs, predicting potential geographic ranges of species using habitat data. It also aids in understanding species-habitat relationships, monitoring ecosystem health, and guiding reintroduction or translocation programs. BCSIR Net Previous Year Questions 1. CSIR Net June 2015. Which of the following is the most suitable method to quantify vegetation cover in a grassland habitat? A pitfall trapping. B-line intercept method. CMIS netting D radio telemetry. Answer. B-line intercept method. Explanation. The line intercept method is widely used to quantify plant cover, particularly in grasslands or shrublands. 2. CSIR Net Deck 2018. While studying habitat preferences of frogs, a researcher records canopy cover, leaf litter depth, soil moisture, and water pH. This approach is an example of a behavioral sampling. B habitat characterization. C mark recapture technique. D quadrat sampling. Answer. B habitat characterization. Explanation. Recording environmental variables to assess habitat quality is characteristic of habitat characterization. 3. CSIR Net June 2021. Which technique is best suited to assess habitat fragmentation over a large landscape scale? A camera trapping. B direct observation. C remote sensing. D quadrat sampling. Answer. C remote sensing. Explanation. Remote sensing provides large-scale spatial data needed for analyzing habitat fragmentation patterns. Let me know if you'd like the next lecture on mark recapture methods, quantification of biodiversity, or home range and territory estimation. You said. Ground sensing method. ChatGPT said. Here is your CSIR Net Life Sciences oriented, high-quality theory lecture on ground sensing methods under the methods in field biology module. This lecture is written in detailed paragraph format with no tables and includes previous year CSIR Net questions with answers and years mentioned. Ground sensing methods. Introduction. Ground sensing methods refer to on-site, terrestrial techniques used for collecting data directly from the environment, especially for studying habitat parameters, species distributions, microclimatic conditions, and ecosystem structure. These methods are distinct from remote sensing, which involves data collection from a distance, e.g., satellites or drones. Ground sensing is an integral part of field biology, particularly in studies involving fine-scale habitat assessments, soil and vegetation analysis, biodiversity monitoring, and ecological modeling. 2. Importance in field biology. Ground sensing methods are vital for collecting high-resolution, fine-scale data on microhabitats, validating and calibrating remote sensing data, known as ground truthing, monitoring subtle ecological changes that may not be visible in aerial images, detecting species-habitat relationships by direct measurement of habitat variables. 3. Major ground sensing techniques. 1. Soil analysis tools. Soil parameters are central to habitat quality. Portable soil probes and auditors are used to measure soil moisture, pH, temperature, texture, and nutrient levels. These sensors are inserted directly into the soil and provide instant readings. Data loggers can record continuous soil condition data over time, revealing how environmental variables fluctuate diurnally or seasonally. 2. Light and radiation meters. Photosynthetically active radiation (PARC) meters and lux meters are used to assess light availability, especially in studies of forest canopies, understory vegetation, or plant phenology. These meters help understand how much light penetrates various habitat strata, which is critical for species dependent on certain light levels. 3. Humidity and temperature loggers. Thermal hygrometers and digital data loggers are deployed at ground level to monitor temperature and relative humidity, especially in microhabitats like burrows, forest litter, or wetlands. These sensors are essential for studying temperature-sensitive species such as amphibians and insects. 4. Canopy cover estimation. Canopy cover is estimated using densiometers, spherical concave mirrors, or hemispherical photography using ground-based fisheye lenses. These instruments provide quantifiable data on forest density, light penetration, and habitat complexity from the ground level. 5. Vegetation structure and biomass measurement. Field ecologists use quadrat frames, point intercept rods, and measuring tapes to determine vegetation height, species abundance, and biomass. Such measurements are fundamental for assessing plant community composition and habitat heterogeneity. 6. Acoustic ground sensing. Passive acoustic sensors, microphones, or recorders placed at ground level can monitor bird calls, insect stridulation, amphibian vocalizations, and mammal vocal activity. These sensors operate continuously and allow detection of cryptic or nocturnal species. 7. Camera traps. Ground-placed motion-sensitive cameras are used to monitor terrestrial mammals and birds. These cameras are triggered by movement or heat and are extremely useful in behavioral ecology and population density estimation. 8. Pitfall traps and drift fences. These are placed on or near the ground surface to sample ground-dwelling invertebrates, amphibians, and small reptiles. They provide insight into species richness, seasonal activity, and movement patterns. IV. Ground sensing versus remote sensing. Ground sensing provides fine resolution, detailed measurements directly at the site and is essential for microhabitat analysis, whereas remote sensing is ideal for landscape level assessments. Ground data often serves as a calibration tool for satellite data to improve the accuracy of vegetation indices and habitat maps. Together, both approaches offer a comprehensive ecological understanding. BCSIR Net Previous Year Questions 1. CSR Net Deck 2020. Which of the following is most appropriate for measuring microhabitat temperature and humidity continuously over a 24-hour period? A quadrat sampling. B soil logger. C thermal hydro data logger. D line transect. Answer. C thermal hydro data logger. Explanation. Data loggers provide continuous readings of environmental parameters such as temperature and humidity. 2. CSIR Net June 2017. A researcher uses a spherical densiometer in a forest study. What is the parameter being measured? A soil pH. B canopy cover. C leaf litter depth. D ambient noise. Answer. B canopy cover. Explanation. Densiometers are specifically designed to estimate canopy cover from ground level. 3. CSIR Net June 2021. To record the activity patterns of a nocturnal mammal species in a dense forest without disturbing them, which method is most appropriate? A mist netting. B pitfall trap. C camera trap. D quadrat sampling. Answer. C camera trap. Explanation. Camera traps are ideal for passive, non-invasive monitoring of elusive and nocturnal species. Let me know if you'd like the next lecture on remote sensing methods, mark recapture techniques, or habitat fragmentation assessment methods for CSIR Net. You said, and remote 1 ChatGPT said. Here is your CSIR Net Life Sciences oriented high-quality theory lecture on remote sensing methods, part of the methods in field biology series. This lecture is written in detailed paragraph form with no tables and includes CSIR Net previous year questions with answers and years mentioned. Remote sensing methods. Introduction. Remote sensing refers to the acquisition of information about the Earth's surface without direct physical contact, typically through the use of satellites, drones, or aircraft-mounted sensors. In field biology and ecology, remote sensing allows scientists to observe, measure, and analyze large-scale spatial patterns in habitat structure, vegetation, land use, climate, and biodiversity. Unlike ground sensing, which provides fine-scale local data, remote sensing offers a broad, landscape-to-global perspective and is essential for monitoring ecosystems over time. 2. Principles of remote sensing. Remote sensing is based on the detection of electromagnetic radiation reflected or emitted from Earth's surface. Sensors mounted on satellites or aircraft collect data across various spectral bands: visible, infrared, thermal, and microwave. Vegetation, water, soil, and urban surfaces all reflect or absorb light differently, allowing researchers to identify land cover types, assess vegetation health, and detect environmental changes. Remote sensing data are typically categorized as active sensing. Systems like radar litter emit their own signals and measure the reflection, e.g., synthetic aperture radar or litter altimetry. Passive sensing. Systems detect natural radiation reflected from the Earth, usually from sunlight, e.g., optical satellite imagery. 3. Applications in field biology. 1. Habitat mapping and classification. Remote sensing is used to create land use, land cover maps that classify forests, grasslands, wetlands, and urban areas. This is essential for understanding species habitat associations and modeling biodiversity patterns. 2. Vegetation indices. Indices such as the Normalized Difference Vegetation Index (NDVI) and Enhanced Vegetation Index (EV) are calculated from satellite imagery to assess plant biomass, canopy density, and photosynthetic activity. NDVI is commonly used to infer ecosystem productivity and seasonal changes in vegetation. 3. Monitoring habitat fragmentation. Satellite data helps track habitat loss, degradation, and fragmentation, which are key drivers of species extinction. Landscape metrics such as patch size, edge length, and connectivity can be derived using GIS software. 4. Change detection over time. Remote sensing provides multi-temporal datasets, allowing ecologists to assess changes in land cover, vegetation phenology, or water availability over seasons or years. This is critical in tracking climate change effects, deforestation, or wetland degradation. 5. Biodiversity and species distribution modeling. Combining remotely sensed environmental variables with species occurrence data enables species distribution models (SDMs) that predict where species are likely to occur. These models are vital for conservation planning and climate change impact assessments. 6. Fire, flood, and drought detection. Thermal and infrared sensors detect wildfires, while microwave sensors are used in flood detection. Drought monitoring is achieved by tracking changes in NDVI, soil moisture, and surface temperature. IV. Commonly used remote sensing platforms Landsat satellites, e.g., Landsat 8 long-term data source for land cover mapping. MODIS moderate resolution imaging spectroradiometer provides near daily global coverage useful for vegetation monitoring. Sentinel satellites from the Copernicus program, high resolution, multispectral imaging useful in ecological studies. LIDAR light detection and ranging offers 3D structural data on vegetation height, canopy layers, and terrain.